Hey guys, how's it going? So in this tutorial, I'll be showing you guys how to pack a cell that contains a surface with a lot of molecules, as you can see right over here. So here I have an hBN surface, or rather a hBN monolayer, and I have packed it with a lot of water molecules, this particular cell. And that is exactly what we'll be learning how to do in this tutorial. So for that, we will be using this web application that I've created, so you can access it at this URL. You can also find a link to it in the description down below. Once you're at this web app, you need to go to the site menu and then find a menu item called pack molecules in a cell, and then go ahead and click on it. Next, you can minimize this sidebar or menu, and here you get this total workflow, like how you are going to do this. So the first thing that you need is a CIF file, that is a crystallographic information file of either the surface or the cell or whatever, like my monolayer. So I need a CIF file that contains this monolayer. Similarly, you also need this XYZ file of the molecule that you want to pack this cell with. In my case, I'll need the XYZ file for the water molecule. So then once you have those files, you can create them using other software or whatever you like. And once you have those files, you can either paste the contents of those files, or you can even just upload the files. So I have those files on my desktop. So what I'll do is I can go ahead and click over here and then upload this CIF file for the hBN monolayer supercell, that is a 5x5 supercell. Once it is loaded, you will see that it will show what it has passed, and you can see that the hBN monolayer looks perfectly fine. You will see the cell parameters and vectors and so on. Similarly, you do the same thing for the molecule. So again, I'll go ahead and click here and upload this H2O molecule XYZ file. Now you will see the structure of the passed water molecule as well, just to see if everything has been read correctly or not. And here is the most important part. So the web app will ask you how many molecules do you want to pack this cell with. In my case, I want to, let's say, pack it with like 20 molecules or 21, and the tolerance distance basically defines how close these molecules are allowed to be each other, at least their center of masses. So next, you can just go ahead and click on pack structure, and it will take a few moments. So you will see this you'll see this fragment where it will tell you where it has added the molecules, and then you will get a success message, and you see that it took around like 3 something seconds to pack this structure with 20 water molecules. And here is the end result. So you can see how easy that was. So that is what I really like about this web app, that everything is so easy to do, so convenient. And once you're happy with your structure, you can just go ahead and download this packed structures CIF file. So here is the CIF file. Then you can go ahead and open it in any software of your preference. And from this CIF file, you can create the input file for any electronic structure software that you want to use for that. There are also other software such as packmol that can do this, but there you have to use the terminal, and it also does not support cells where the angles are not orthogonal to each other. So the lattice vectors need to be orthogonal for packmol to work fine, while this app will work for any kind of cell. So yeah, so that is it. I hope you guys really enjoyed this tutorial and found it useful. In case you did, then don't forget to hit the like button and subscribe to my channel for more videos like this. Thanks for watching and have a great day.